Yes. Tell
Hello, hello.
No.
No. Hello.
No.
Hola, 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 hola. Ah, no sé si entré per Zoom.
Però per Zoom jo estic veient aquí.
Ah, vale, vale, vale.
Sí, ho estic veient. Mira.
Vale, vale. Okay, good. So, good morning, everyone. I am Jordi Garcia-Ojalvo, and first of all, I'd like to thank you, Seb, for putting this thing together and for persisting on this after so many months. And finally, it's happening in a very strange way. So I hope, I don't know, I am trying to be mindful of the people that is online. I don't even know if they can see my faces or anything. Okay, Carlos is saying that it sounds a little distorted. Maybe it's me. Okay. Is it better now? Carlos? Yes? Vicente, great, thank you. Okay, I want to be mindful of the people who is online, and this is a rehearsal for all of us. And I don't know what's going to happen when I start teaching in a few weeks, so I want to rehearse this because I think we're going to have to do these hybrid things for a while. And I want to be mindful of the people that is watching online also. I don't know what's going to happen, so I apologize already for all the things that are going to go wrong. That's the first thing I want to say. Thanks again to Giuseppe. And anyway, let's say, I'm sorry I was not here in person yesterday. I was able to see Tomas's talk, which I find, but not unfortunately Mayra's nor Imma's. Anyway, so this is what, according to the website, I should be talking about, but it's a bit doesn't mean anything because it's a whole topic of the school, if you will, no? So anyway, I'm going to be explaining exactly what this title means in the next few minutes. So we have seen already very nice examples of dynamical processes in populations from yesterday, no? Both at the level of diseases and disease propagation, also at the level of population in general. And today I would like to continue talking about biological systems, but at a much smaller scale. So instead of talking about populations of organisms, I would like to talk about an organism by itself, and then I zoom in, and then I'm going to talk about cells, okay? And why? Because, well, most of you, I imagine, are mathematicians or physicists, I guess, in the audience, except for some, you are not, yeah, so anyway. Oh, Giuseppe, no, Giuseppe is saying, I'm going to try to be mindful of the chat. On the other hand, if somebody has a question who is watching online, I guess maybe you can tell me. So, because please interrupt me at any time, both here and through Zoom. Just say something through chat, and I'm going to ask Giuseppe if you want to ask a question. Okay. So anyway, let me go back. So we are all, most of us are quantitative people, mathematicians or physicists. Why should we focusing or why should we be interested in biological systems? Yesterday you saw it very clearly. Biological systems, populations are very dynamical. There are lots of things happening in time. And as I was telling you a minute ago, I would like to focus more on a single organism. And it's clear that when you look at a single biological organism, time it is important. Time matters. Okay, this is an example, the first that you could think of, the development of an organism, a multicellular organism, a mammal, a mice, or ourselves, starting from a single cell. I really love this slide. I use it a lot because I think it's impressive, right? We start, all of us, from one single cell, which comes from somebody else, by the way, so cells are not invented, so we are all coming from the one original cell many millions of years ago. So we start from one cell, and then this cell starts proliferating in the same way that populations proliferate, as Thomas was saying yesterday. And then at some point, the cells start to decide to do different things a long time. So as time progresses, things change. And things change in the sense that you have all these cells that start to make decisions. Some cells are going to become blood cells, muscle cells, skin cells, brain cells, heart cells, okay? And this has to happen in the right order, at the right time, and eventually, we end up with an organism which is, in my opinion, an amazing example of spatiotemporal organization, self-organization. Okay? So it is clear that time matters. This is a process, a long time, which is very well defined. In fact, it's amazingly well defined, right? I mean, my PhD advisor always tell me, and we were not working in biology at that time, but he was amazed that we always, all of us had 2 eyes in the right places and 2 ears, and how can this happen when, as we will see in the third lesson, hopefully, there's a lot of noise and stochasticity, right? It's really amazing that we have this, this organization, okay? And this all happens through time. Things have to happen in time. Cells have to decide at the right moment to become the right type of cell, and depending on the time and also on space, but I'm not going to enter too much into space, okay? I just want to talk about the temporal aspects of this thing. So it is clear that biological single organisms are really very dynamical objects. So this means that somebody who is working on dynamical systems like us, then we could say, okay, let's try to see if we can do something with it, okay? That is one example. We don't need to go to a single organism. We can get one of our cells in our body, of which we have billions, which is, for instance, you take a little bit of blood, and you get out of the blood white cells, white blood cells, the ones that are responsible for our immunity, right? So important nowadays, no? And you may know, I don't know if you know, does somebody know this movie? This video, it's in YouTube. I will post these slides, so I will make them available so you can take a look at them later. This movie is really nice. So what you have here is blood. You have here all these circular things which are red blood cells, the ones that they carry the oxygen and so forth. And then this is one of these white blood cells, the one that we provide immunity, okay? So this is a neutrophil. And this is a very well-known movie, which is already very old, but it's one of a hit, it's a hit in the internet, because this is what happens when this cell encounters a bacterium in the blood, okay? I don't know, I don't remember this, I think this is, I don't remember, Salmonella, I think, I don't remember what you can look at it in the website. Anyway, let me show you the movie. And what you can see is that single cell, as time evolves, is entering into a race, and it's very nice and fun, and you can watch it, and you don't need to think about the biology or science of it. Just, oh, look at this little thing going after the very smaller thing, right? And it's just nice. But of course, there's a lot of interesting processes, biological processes involved in this, right? The neutrophil, and eventually, of course, it eats it up, right? So this thing here has to detect, has to have some kind of chemical sensing that tells that the bacterium is there, right? The bacterium also has to have some chemical sensing that tells that the neutrophil is there. It has to run in the opposite direction, and then this thing has to reorganize. This big cell has to reorganize its entire skeleton so that it starts crawling in the direction, moving in the direction of the bacterium, and finally, then it has to phagocytize the whole bacterium. So this is an amazing example of dynamical process again. Okay, so you don't need to go into a whole organism. You can go into a single cell, right? But we don't even need to go into a single cell. We can even go farther down, and I'm going to keep going farther down because now my question is, how do cells do this? How do cells do this? What is the main biological component that makes cells do stuff? And I'm going to ask, I don't know if you know, because I don't know how much biology you know, and you may know a lot, and you may be bored about what I'm saying and what I want to say. So I know, what is the main biological component that cells use to do stuff? Can somebody tell me here or in Zoom? Somebody? What is the main component? How do cells behave? How can they? This is an amazing thing that matter can do. There's no magical or mysterious thing that provides these things with the drive to do this. There are little objects. No? Okay, at the end of the day, what's really, the cells are mostly using, and Santiago will keep me honest here, I hope, because, okay, I'm going to show you a video. Of course, you cannot watch it. You cannot watch these things in real life, so what you do is animations. And this is in the internet. This is a project from Harvard, which is called BioVisions, which is called The Inner Life of the Cell. You can go and look at it. And in this particular case, their focus is on neurons. I'm not going to enter into this. I believe the cells in our brain, and you can see here the axons, the soma, the nuclei, the big part of the neuron. These are the axons. And then you see the fibril. I don't care about this right now. You see the membrane of the cell, which all these things, and then you go through one of these little pores that cells use to bring stuff inside, ions, for instance, and then you go inside, and what you have inside is this. These big molecules, in which geometry, from my opinion, geometry and thermodynamics is what's important, because these molecules have very well-defined shape, which is just due to the way that the different atoms in it in them are configured, and then these molecules are moving, and they have to find each other, and they have to connect with each other due to thermodynamics. And then when they connect with each other in the right way, they bind, things happen. Things happen, such as, for instance, they can catalyze reactions, chemical reactions, to transform something in something else, and thereby maybe you can generate energy. There are ways in which you can, of course, the cell has to get nutrients and generate energy. It has to move, and you haven't seen it earlier, but one of the proteins I was showing you earlier here was actin, which is part of the cytoskeleton, the so-called skeleton of the cell, which is what's moving. When the cell is moving, it has to create more of this in one direction rather than in the other. It's an amazing dynamical system problem. We could look at this video forever. The important thing I want to tell you is that the main component, in my opinion, is proteins, which are very complicated three-dimensional structures that when they bind in different ways, they produce all this kind of emerging behavior, okay? That's the whole thing. So the question is, what proteins, this is what the biologists tell you, is what proteins are the cells expressing and at what times? So the important thing is the expression of a protein. What does it mean, expression? So first of all, how do the proteins appear? Usually people tell you, you have to eat your nutrients, no? Nutritionists and mothers and fathers tell you, you have to eat proteins. You have to eat your proteins, right? Some people tell you have to eat meat because this is what is the source of proteins. It seems as if the proteins we are eating, we have to eat them, but we don't have to eat them all. We can create them ourselves, right? And we create them using this type of process, which was called many years ago the central dogma of molecular biology, which tells you that the proteins are coming from our genes, and that is why our genes have become so important, no? So we know our genome, we know the list of genes that we have in every one of our cells. These genes are going to be what's called transcribed into mRNA. Almost anybody knew about mRNA before. It's very nice that now, thanks to the vaccines, everybody says it's an mRNA vaccine. Well, I think that you agree with me that at least one positive aspect of this whole crazy thing is that now everybody knows what an mRNA. I mean, 1 year and a half ago, I gave this talk to people who are not biologists, and they don't care. Now everybody knows what they recognize the mRNA. Anyway, mRNA here is just an intermediate between the gene and the proteins that do stuff. Of course, Santiago will tell me, no, but the mRNA can also do stuff, and I agree, but I'm oversimplifying, okay? So anyway, this is the way in which, in principle, this is the process that's called gene expression. So the genes that are here are going to be expressed into proteins, and depending on how much they are expressed, you're going to have more or less of this protein in time and in space. And that's the whole thing. And then, of course, the proteins can diffuse and things happen in space anyway. So this is the flow of what we could call it. I'm not going to call this the central dogma of molecular biology, which is very dogmatic. So I just call this the flow of genetic information. By the way, if you want to see the process in real life, this is it. This is a picture. It's very hard. I told you earlier that you have to rely on simulation and animations, but this is the real stuff. What you see here, this thing here is DNA. Okay, look at this is electron microscope image, I think. Okay, so just see the mRNA, sorry, the genome, the genes, and oh, wow. Okay, what happened? Maybe it's me. How can I solve it in some way? I don't know, Jordi. Maybe I can do this. This changed. Wow, I have control. I have the power. Okay, great. Anyway, so let me. Okay, so so if you have never seen the genome, you've never seen a gene or a piece of DNA, it's here. And all these things here, it's hidden, but this is the mRNA molecule, okay, that has been produced. So here there was that's called an RNA polymerase. I'm not going to an enzyme, a protein that it's making the transcription, and then you have the molecule, and then these little blobs here are so-called ribosomes. Other enzymes, very big, very important, very amazing molecular machines in themselves. What they're doing is they're translating the mRNA into the protein, and the protein we cannot see. The protein we cannot see. So this is a cartoon of this. This is a nice article, by the way, very old already. This dates me. And you can see the DNA. This is the RNA polymerase. This is the mRNA. You have all these ribosomes, and these are the proteins being produced. This is a thing. This is true in bacteria. In our cells, we have nuclei. Our cells have nuclei. We have different kinds of cells, and in our cells, the process of transcription and translation are separated in space for whatever reason that I don't know. I'm sure Santiago knows. Okay, so I'm sorry if that I'm always. I apologize. So anyway, but this is a bacterium. In a bacterium, these things happen in real life, and you can see it here. Okay, so far so good, I guess. Now, of course, if this was all there is a big problem here, right? Because if that's all there is, this means that once we have our genome fixed, this means that our proteome, what we will call the proteome, the list of proteins that our cells produce will be the same. So for instance, all the cells in our body have more or less the same genome. There are some mutations that we can quantify how many, okay? But basically, all of the cells in our body have exactly the same genome. According to this picture, they would have exactly the same proteins. So this means that we will not be able to have different, oops, I was giving out the answer. We will never be able to have different types of cells. For instance, in blood, we have red cells, white cells. Here, these are neurons in our brain. There are many more. No, we have around, and people say that we have around 200 and something cell types. Nowadays, people are saying that maybe we have thousands because they are discovering new things. So we are jumping from 200 cell types to thousands of cell types, right? So how can this happen? How can a cell in our skin and a cell in our brain, which have exactly or almost exactly the same genome, be so different? And now I'm asking you, what do you think is missing? And I know that Bly knows, but anyway, and maybe somebody else who's not Bly. Regulation. Who said it? Oh, okay. Thank you. So, what's your name? And let me get to know. Stefano. Great. Regulation. Regulation. Thank you, Stefano. Have it here. Okay. I'm going to. Okay. Yes. I'm sorry. I don't want to. I cannot avoid it. I'm already entering into teaching mode in 3 weeks. Anyway, thank you, Stefano. Yes, regulation, right? I mean, the amount of protein that you are producing is regulated by whom? Well, somebody might say also by external signals and whatever, but not directly. Usually this happens because proteins are producing the regulation, okay? So now this completes the picture, and this allows this type of things to happen, and later we can discuss about this if we have time. Anyway, so now, of course, this is a single gene and a single protein. It seems that it's activating itself. We'll come back to this in a minute. This is interesting in itself, in my opinion, in my very reductionist opinion. That's already interesting in itself, and we will focus on this in a minute. But of course, what can happen is that you can have a few of these genes producing proteins and interacting with each other. And this leads to what we could call gene circuits, right? So we'll come back to this. That's what we're going to be analyzing today and tomorrow. I mean, we know that we have thousands of genes in our cells, right? We know that we have, I should have asked, but maybe we know that we have, I don't know, 20,000, 25,000 genes or something. Then these genes, every one of these genes can encode for more than one protein. I'm not going to enter into this. So you can have millions of different types of proteins. So the situation can be very complicated, and I'll come back to this in a minute. But I just wanted to define an object that is very interesting in itself, and that we can analyze it mathematically with the techniques that we have learned in this school. Okay, and that's what I call a gene circuit. And for me, a gene circuit is a collection of a small number of genes and their proteins that are interacting with each other. For instance, this little thing here denotes the gene, a piece of the DNA that's controlling the production of this gene, and then you produce the protein, and the protein activates itself or something like this. You have activations, you can have also repressions, and I'll come back to this in a minute. This is what I define as a gene circuit, okay? So finally, I got to the list of things that I would like to talk about in these 3 days. I usually go very slow, as you can see, so I'm not very good at maintaining time, but we'll see what happens. I wanted to talk about 3 things. First is a general introduction to why I think it's important, gene circuit dynamics. So it's not only important, it's not only nice because we can do something with it, and we have all these tools that we are learning, and you already, some of you know already very well, and we can do things, and they are important biologically. I would like to discuss this today. That's what I would like to discuss. And then I would like to give you an example, which is biased. Unbiased. I'm going to give you an example of how we can apply the tools of the dynamical systems to understand a specific biological process. And I am using an example of something that we worked on quite a few years ago. It's old, so nobody would class. This is not a conference, so it's not. I'm not telling you our latest discovery. I'm telling you something very old. which I think it's a good example, in my opinion, just to show the power of dynamical system analysis in these things, okay? And I will show you how to dissect, if you will, a genetic circuit using dynamical systems approaches, okay? And finally, I don't know how much I will have time to go into the topic of noise. You have already seen that these proteins are moving like crazy inside the cell because we are at 300 Kelvin, we are not at 0 Kelvin. So thermal fluctuations are very important in these large molecules, and they're moving like crazy. How do they manage to work in a consistent manner in spite of this huge amount of noise? Okay. Now, I don't know how do you distribute the material. I don't know, Thomas, if you have already. I don't know. You send slides, and then the organizer sent an email, and I didn't ask. Well, I don't want to do it, and I'm like enough because I don't want to do it before because I want to keep the surprise. But I will do it later. So that's one thing. Another thing is I have already placed them in a GitHub repository. I know that we shouldn't use GitHub repositories for this, but that's anyway. I created a repository with only material for the course. Okay, so this is my GitHub account, and there is this CRM course. So you can, and if you want, either you take a picture of this thing with your phone, or which I don't think anyway. But somebody is doing it, so thank you. Or you just take this Jordi and the CRM course. So I will put material there, but I will also email you if you want. The problem is that the PDFs are large. So in any case, I would put the PDFs in this website, and that's it. Okay. So this is the thing. This is what I am going to be using. So now let's start. I don't know if you have any question or comment. This is supposed to be. I mean, we decided that this is not a conference. So the advantage of a school is that we can discuss and talk. And so that's the point. I mean, I'm not. It makes no sense to just show you the beautiful thing that we have done yesterday. So I think and this also includes dialogue. So please, if anybody has any comment here or on Zoom, so far so good. This is nothing. I haven't said anything complicated, but anyway. So let's focus on the first part. Why is this important? What can we do with it? How do we model this type of circuits? Which you'll see that these are the same tools that we use for population dynamics, right? So anyway, so let me go back to showing you the picture of a genetic circuit. No? So far so good. Now, mathematicians, biologists have been showing this type of pictures for years in their papers. No? Because they do a lot of work, biochemical, using many techniques to determine these interactions. Well, this one is trivial because this is the gene producing the protein, but the fact that this protein is activating itself, or the fact that this protein is repressing or inhibiting this one, it's hard to see, but you can do assays, and people do it. People spend one full PhD, that's what I would like to think, just to find out maybe one of these arrows. And then we say, oh, we don't want to be so reductionist, I want to be thinking, no, but it's very hard work and it's very important. Okay, so you can build these things. So then biologists will show you these figures in their papers because they come from the summary of the results. And then my experience is that they are amazingly intuitive. When they have a figure like this, they can tell you, oh, look, this gene, this protein expression is going to be going up, but this one is going to be down. They have an amazing intuition interpreting these things. But my point is that this has a limit. Okay, there are many complications that prevent you from determining what's the outcome of this behavior, what's the behavior of this dynamical system. Okay, proteins going up and down and whatever, starting from an initial condition. So, I mean, look, you have feedbacks. You have a positive feedback. We'll come back to this later. You have a positive feedback here. You have a negative feedback here. This is a clear example. And I'm not sure if I will have time to finish today's talk with this example, or I will start tomorrow's talk with this example. But here you have a situation in which you have a positive feedback combined with a negative feedback. Which one wins? I mean, I cannot imagine an intuition that tells me this just by looking at this. Okay, so that's my first problem. We have feedbacks. It's complicated. The second problem is that these interactions are usually nonlinear. Okay, and that's what now we are already getting into things that are interesting to us because we are doing dynamical systems. We are doing nonlinear dynamical systems because linear dynamical systems, what? Why? Right? So but thankfully for the scientists and mathematicians, the interactions are frequently nonlinear. I mean, the response, the production of the protein with respect to this thing, which is called a transcription factor, this protein that controls the expression of this gene, it's called a transcription factor. So if I graph this, this is not a linear function. Okay? So now the first problem is there are no linearities. And we know what happens when there are no linearities. We cannot solve many things many times with analytically. That's fast. That's for mathematicians and physicists. But the intuitions is very hard because you don't know. There are thresholds and things. We'll come back to this in a minute. That's the first problem. The second problem is that, as I was saying earlier, I just showing you 2 genes. But there are not 2 genes. There are thousands. No? So this is I went to a website that probably it's over because these things change very much. And this is maybe more than 10 years old. I entered into this database and I generated this, which is the so-called gene regulatory network for pluripotency in stem cells. And okay, what does it mean? I'm not going to enter into it. Detail. Stem cells are cells that can become anything. Okay, they are powerful, super powerful, and pluripotency means that they can become anything. So what are the proteins and genes involved in controlling this process? I go into this website and I get this. These are all the genes, or when I say genes, I'm going to say genes or proteins interchangeably for the moment because one gene, I'm going to assume one gene is equal to one protein, which is not the same, it's not true, but anyway. So you have all these genes here connected in a very complicated manner. I don't call this a circuit. This is just me. This is a network. Okay, we all agree. This is a network. And then we can study the characteristics of this network or whatever, the statistics of the degree or whatever. It is clearly a network. Okay, but of course, the problem is that then you have a lot of nonlinear interactions in this network. So you have to determine the dynamics of this thing. So that's what I call the challenge number 2. Okay, and finally, the challenge number 3 is that living processes are dynamical. Cells are moving. Cells are moving. And are interacting, are changing partners, and situations are changing, which means that these networks, and this is a very bad simulation I did of a network in which the interactions are not active all the time. When you open a textbook or a paper and they show you something like this, it's very misleading in my opinion, because you are thinking that these things are active all the time, but that's not true. So, I mean, these things are moving away. This is a dynamical system. So you have a nonlinear dynamical system with a huge number of degrees of freedom. This is my challenge. This is the challenge that we have here, one of the challenges, if you will. Okay? So anyway, this seems hopeless. I have to tell you that biologists have neglected this particular aspect for many years simply because it's hard to gather dynamical information. You have to on purpose, you have to track your biological system. You can do movies like this, and the technology to do this type of things is now very well established. But even so, people are not doing it in the way that 10 years ago. I mean, this technology, I would say, let's say it's 20 years old, more or less. And I would say that by now everybody would be doing movies. Okay, I would say, but if you look at the literature, not so many people are doing movies. For several reasons, there are limitations of how many degrees of freedom you can measure at the same time. I'm not going to enter into this. This is a real problem, but okay, there are other things, there are many problems in life. But the scientific community has decided that this is not the way that we want to go. And in reality, even though microscopes are available to do this type of thing, sometimes the microscope crash. It's very hard to do a very long thing. The movie, somebody, you could say, oh, this is not maybe in realistic conditions, even though you can do this type of movies in vivo. And we have some results with some collaborators recently about development of mouse, in which they have amazing movies in vivo. Okay, but people are not doing this so much. But in the past, it was impossible. So in the past, what they were doing is they were taking pictures, snapshots of a biological system. They take a picture, they freeze time, and they see what is the content, how many proteins are being produced in any given time. And from that, then you have to try to extract the dynamics, and that's the complicated thing, okay? So, but I think this is still relevant, and I want to give you some examples now. Why is it relevant? First of all, I'm going to show you that there are many examples in living systems in which dynamics is important. I don't know, maybe I can do a survey. Things that come to mind, examples of dynamical processes in biological systems that come to mind. Does somebody want to say an example? The risk with this, I always do it with the students, but I know, and then they never answer what I want, but of course it's very important. It's very difficult. I'm not going to blame anybody because, of course, you cannot read my mind. Let me give you the first example. Oh, what? Well, for instance, no, you are going to give examples of microscopic. Of course, let alone population dynamics is a clear example. When I'm talking now about cells and cells and viruses, it's called like this. And definitely there's a lot of dynamics, and you will be talking about this when you talk about quasi-species and so forth. So that's one example. But I want to give, this is it's important. I think it's very important because it's right now nowadays it's becoming something very important. But something that is very clear, it's in all our lives is in 4 hours you're going to be hungry. 3 hours. Now, you are not hungry because you have, okay, you ate breakfast, but it's not because you have a bottle that is becoming empty and, oh, now I don't have enough food, I'm hungry. No, you are hungry because it takes you, because you can tell time, because you can tell that it's 2 p.m. and it's time to eat, because we have clocks in our bodies, right? We have clocks. In particular, in our brain, there is a little area, I'm not going to enter into this, hidden inside our brain, in which the cells, the neurons, are fighting with a periodicity around for 24 hours in the presence of light-dark driving. It's exactly 24 hours. Of course, without light-dark driving, it's not exactly, because it would be amazing that the cells would be keeping a rhythm exactly a 24-hour period. So it's slightly different, but this oscillator is entrainable. So I don't need to tell you about this. So basically, we have oscillators that can be entrained by the light-dark cycle. And you can see for 14 days, the neurons are fighting periodically with a period of 1 day. If you remove light-dark illumination, this thing keeps running. I'm going to go show you this. This changes our hormone contents. This changes the state of our immune system. And one underappreciated, in my opinion, thing, and you can tell me, is that people don't care about what time of the day we ingest therapeutic things, drugs, for instance. I don't know, maybe we could have a discussion. I wonder if there is data on what is the outcome, and probably not because it's very difficult. What is the outcome of the vaccine, of the COVID vaccine, depending on the time of day that the vaccine was taken? And I would love to see data, now that we have millions of people, just if they recorded at what time of the day the vaccine was introduced into our bodies. I wonder if we would see any pattern. This is pure discovery research. I just want to see if there is any pattern. My hypothesis is that there should be patterns because the concentration of the different types of immune cells in our body is oscillating with a 24-hour period. Our temperature, our pressure, and therefore the number of heart attacks oscillates along the day. Okay, it's more probable that heart attacks happen early in the morning. This is a fact, and this is because of our circadian rhythms. So that's nice. You could say, oh, look, we have some cells that are like clocks inside our brain, but it's not it. All the cells in our body, because the clock is sending hormones through the whole body with a period of 24 hours. So this means that every single cell in our body is subject to an environment, receives signals that are varying with a 24-hour period. So these cells are driven, temporally driven dynamical systems. So we have a driving with a 24-hour period. And this is not enough. This is under study. It's hard to study, but I'm telling you, I don't know, maybe I'm wrong, if there has been a study posted somewhere in which they look at the effects of time, which is called the chronobiology of the SARS-2 vaccine. I wonder. It will be nice. Yes, Thomas. Yes, of course, please.
If you can quantify the different types of neurons in our body, which one of them have an intrinsic clock and rhythm, and which one can be trained by our night, our waking things?
Yes, that's a wonderful question. There is this concept of peripheral clocks. So I don't have I have some more slides in another talk because I'm not there is a concept of peripheral clocks. Our liver has a peripheral clock, our kidneys, our muscles. So we have clocks because all the cells in our body have the same components of which I will talk in a minute. This clock is driven by proteins and genes, by these circuits. And all the cells have the same genome. Of course, some of them are not going to express some key products, and that is why most of these peripheral clocks are not self-sustained. So we are concentrating on this because these cells are self-sustained oscillators. The rest have only maybe pieces of the component, or they don't have the proteins expressed at the right levels, and this means that they are not self-sustained, they are damped oscillators. And most of the clocks in our body are behaving like this. Now, there are studies. Of course, it's always difficult to study these things in vivo and quantify directly, but this happens. So we are a collection of peripheral clocks driven by the clock in the suprachiasmatic nuclei, which is this thing that we have in the brain. And we have, this is a problem of synchronization of dynamical systems. Okay, so synchronization of oscillators. The liver is one oscillator, the heart is another. Well, and let take the heart, by the way, is another oscillator with a period of 1 second, right? But I'm not going to enter that. That's another example that I don't even have here. Okay. Anyway, more because I'm talking too much already. Another example that maybe you haven't think of, but it's very important, is the cell cycle. When our cells proliferate, and some of the cells in our body are proliferating continuously, which are, for instance, the cells in our skin, in the interior of our gut, cells that usually are very much affected, or immune system, cells that are prone to getting cancer, because whenever there is a proliferation of cells, then you can get mutations, right? So you have these kind of things in which a cell becomes two, and then these two become two more, and so forth. And this is a clock. The proteins that dictate the division have to oscillate, and we know by many mathematical modeling studies that this is also a self-sustained clock, differently in different species and so forth. Okay. Now, before I continue, you see here very nice colorful plots, red and green and whatever. I'm going to show you what it means because I'm going to use it. I'm going to use it quite a bit in these 3 days. Okay. What does it mean when you see red and green? Okay. By the way, red is some protein that simply tells you this protein is expressed during this phase. These are phases of the cycle. The cycle is divided into 4 phases. During this phase of the cycle, this red protein, let's call it the red protein, is expressed. You can see that this is a dynamical system because that protein is only expressed in this phase. And green is another protein that is only going to be expressed in this other phase. So if you are able to, in time, know how much of this protein exists in the cell, then you have these beautiful pictures. How do you do it? I'm going to tell it once. I think it's useful to know. The way of doing it, and it's very standard nowadays, a Nobel Prize was given a few years ago for this, for the components, which is you use what's called a fluorescent protein, okay? So a fluorescent protein, you can get fluorescent protein from many different species, some species in the sea that are intrinsically fluorescent, so people got these things from this species, so these are not human or mammalian proteins, but you can put it in the human cell or in a mammalian cell, and then you fuse them to our protein of interest, the red or the green or whatever. So you want to track in time how much of this protein is being produced and where it is, for instance. So what you do is you fuse this protein to the fluorescent protein, you pray, well, sorry, this is not very scientific, but scientists do it all the time because you're doing an experiment and you have to pray that it works, because otherwise you have lost 1 month. Anyway, what can I tell Blair, right? Anyway, you put the two things together. You can do it with, you can cut and paste the DNA. Basically, you work at the level of the DNA. When the DNA is expressed, then you get something, this an extended version of the real protein that includes this little thing tagging along. And then you pray that this little thing is not affecting the function of this large thing. Well, they're not necessarily large and small, but so of course, this only works if this thing is somewhere in there and it's not affecting the main function. That sometimes doesn't happen, because if the main function requires some area of the protein in here, you are blocking that the thing, and then it's not going to work. So you have to look that the protein is viable and is functional. But if you do it, then you get these beautiful pictures, and I'm going to be using them later. This means that you can put it in a microscope and you can follow them. Of course, here you can see the problem. You cannot track 20,000 genes or whatever, or the corresponding proteins. First of all, because you don't have enough spectrum, right? Because these things are fluorescing with in a certain area of the spectrum, and the spectrum is limited, as in radio, right? So you cannot have infinite numbers of radio. Well, nowadays with the internet is, but anyway, you cannot have infinite numbers of radio stations. It's the same thing. Okay, so we are limited. We can have 3, 4, whatever at the same time. That is, I think, the main limitation, and that's the excuse that scientists have used to say, I'm not going to do my movies, which I think is a mistake. But anyway, maybe it's only me. Okay, because we don't have this information anymore. Okay, so this is another example, the cell cycle. And now I'm going to show you more examples in which we can track these things. The location of, for instance, this movie here. This became very famous. This was published in 2004. And since then, it has been reproduced many times. What you're seeing here in red is one particular protein, which is called NF-kappa B, NF nuclear factor, whatever, I don't know. It's a transcription factor that is very important for the immune response of cells, okay? For apoptosis and whatever, it's very important for the immune ability of cells in our body. And you can track activity, meaning since it's a transcription factor, it regulates the gene, it is important that it's inside the nucleus, because this is where the transcription happens. So you can see that the protein is inside, outside, inside, outside. You can see that this is the nucleus, and the other thing around here is the cytoplasm. It's outside of the nucleus, but it's still inside the cell. And you can see that it's amazing, if you will, because almost all the copies of this protein decide at the same time to enter the nucleus and then to leave after a while in a synchronous manner. This is also a problem of synchronization, if you will, because you have all these little molecules here. You have, I don't know, 100,000 copies of this protein inside the cell, and almost all of them decide to go inside the nucleus at the same time. They do their stuff and then they go out. Okay. So anyway, this is in response of a cytokine. So it's a typical immune assay, if you will. And by the way, if you ask me what, yes.
So the protein comes in and out.
Yes.
It's not about the degradation or production.
Well, that's a great question as well. Of course, degradation is also involved, right? So, but it's really true that it's being translocated. I mean, these proteins, if you think about it, the life of a transcription factor. What is the life of a transcription factor? The mRNA is produced inside the nucleus. This mRNA has to go out. Outside the nucleus, you have the ribosomes. You produce the protein, and this protein now, in order to regulate the expression of the gene, has to go back in. Okay, so it has to. I mean, there are methods, there are biological processes that regulate what's called translocation of these things inside and outside of the nucleus. And the amazing thing is that it happens synchronously. Of course, there is degradation. To take into account dynamically, this is also important, and we will have it in our models. Yes, thank you. What is your name? So we have Stefano over there, and you, Mark. Great, thank you. And this is not just for me to remember, sorry. Okay, so that's another example. If you ask me what is the period, first of all, I think nobody knows, if I am not wrong. Hey, ¿qué tal, Tomás? I think nobody knows what is the function of these oscillations. Please correct me if I'm wrong. Nobody knows why the immune cells, and people have looked at it in many different cell types. This was in mouse, but people have seen it in humans and whatever. And just if you put the cells in the right conditions, which maybe they are artificial, I don't know. I don't think so. Nobody knows what is the utility, if you will. The period, what is the period? Let me remind you that we're talking, I haven't talked about this, but this was 24 hours. This thing here depends a lot on the type of cell. The cells in our body could be also 10 to 24 hours. Bacteria usually, I don't know, 20 minutes to an hour or whatever, or 2 hours, depending on the conditions. And this thing here, inside our bodies, it's around 2 hours. And it's quite robust. We have this oscillation with a 2-hour period inside our blood. If you hit the cells with the right amount of cytokine, and cytokine is one of these molecules that are signaling for the immune system. In this case, it's TNF-alpha. Why 2 hours? Well, I don't know. Maybe. No. Yes. Yeah. Yes. Yeah, that's another interesting thing, right? Thank you, Santiago. It's true. When I was talking about synchronization, I was talking about synchronization at the molecular level, all the proteins going in a single cell. But it is true. You have to think about this. The cells are in the same conditions, and the phases are not so these are not synchronized. So they seem to be uncoupled, these oscillators. If you interpret each one of the cells as an oscillator, they seem to be uncoupled. As far as I know, I don't know if there are examples of NF-kappa oscillations in tissue or something in which you see oscillate about tissue. I mean, this makes no sense. Yes, for sure. That's also true. They are moving around a lot. That is true. The space is very important. I agree. That is true. This is completely artificial in that regard, right? You put it on a plate. The cells are moving like crazy, and then they don't have enough. Maybe they don't have enough time to be in touch, so that they synchronize. And maybe in real life, in the thymus or something, I don't know, they are more in contact with each other. The density is higher, and maybe they are synchronized. We don't even know. there are reports of immunologists that tell you that you do have, well, there is knowledge of oscillations in the immune system. Ultradian, this is called ultradian because it's faster than the circadian. So there are examples of different periods macroscopically. I don't know if this is, maybe this is an artifact or maybe, I don't know, but this is not known. I think that's why I'm telling you that I think it's important to track these dynamics. Of course, it's very difficult to do it in vivo. This I agree. So we can only do it in a bunch of, I don't know how. Yeah. Then since you have a lot of power in imaging, yeah. But I have the feeling that people have moved on. I don't know if you agree with me. I have the feeling that people have moved on, and now they are all into this craze of single cell RNA-seq, cell atlases, and whatever. Which you do? Sorry, I don't want to antagonize. No. Well, I'm saying it is important, but what we shouldn't do is we should all work on this, and then we only get funding if you work on atlases and in classifying cells. I think, and people have run away. This is the way funding in science, I think, works. Everybody's pursuing this thing, and the agencies are also giving money for specific things. AI and cell atlases. That's what you have to do nowadays. Artificial intelligence and cell atlas. Come on. I'm too old, I think, to do this. Well, I understand. It's very expensive, and you have to decide what to focus your attention on. This is true. This is not mathematics in which you just use your pen and paper. And of course, we, from a theoretical point of view, we need this, but this is very costly. So I understand. But I think it would be good to bet to hedge our bets, to todo los huevos, all the eggs not in the same basket. Okay, so, but anyway, again, that's my opinion. Anyway, sorry, let's I don't know, we have 1 hour and a half. I don't know if I should stop. Okay, great. So I'm going to continue if you're not tired. All right. Okay. So far it seems that everything is oscillations, which is nice. We love limit cycles, right? We love limit cycles. So it's okay. But it's not only oscillations. Okay. Let me show you, for instance, this. I'm using always very old papers because these are the ones that I mean, I'm old, so that's it. So this was published, I don't know, 17 years ago. Okay, this is the group. It's a group of Uriah. I mean, this is a dream team. No, you have Uriah Alon, you have Michael Elovitz, you have Arnold Levine, and you have Galil Lahab. So anyway, this is a very old paper. We have 4 big shots right now working together at that time. Okay, and what they saw is something very simple. They use these techniques I was telling you, these fluorescent proteins, and you have, I don't know, it's very hard to see, but here you have 4 cells. You have 4 cells. I'm sure that on the Zoom, probably it's very hard, and I am sorry, I don't know if this is, but you can tell me, but it's probably impossible. But anyway, so there are 4 cells here. This is a bright field image. You can see the cells, and then what happens is that you take these cells and you put you fry them with gamma rays. Okay, you fry them with gamma rays, which are going to produce mutations. They are going to produce mutations in the DNA. The cell is going to detect that there are broken parts of the DNA, whatever, and of course the cell starts trying to fix it because our bodies are amazing. That's another amazing thing of the biological system, that we self-correct. Amazing self-correcting device. The immune system is an example, but inside the cell, when some DNA molecule breaks, some guy comes and tries to fix it. And this guy is regulated by the protein which is called P53. Okay, P53 is one of these famous anti-tumor suppressor genes. I think the discoverer is Arnold Levine, so that's why this was done in P53. And since then, Galit, as you will see later, I will show you another example. She has been working a lot on this system. Anyway, so you fry the cells, you break DNA, and then you track these 4 cells. And you can see this one cell here. It goes green, which means that it's expressing P53. P53 is bound to a fluorescent protein. So P53 starts to appear, and it disappears forevermore. So that's the time trace. You have 9 time traces. Okay, no oscillations, 1 pulse. This sister cell here, for whatever reason, goes once, and then it goes again, and then it goes down. Tick, tick. So it does twice. Okay, so first of all, again, going back to the idea of Santi earlier, there is no synchronization. Different cells are doing different things. Of course, it could happen that maybe this cell, I think what's believed is that these pulses are attempts to try to fix the problem. And if the problem has not been fixed, then it tries again. So you might think, oh, maybe this cell is more badly damaged, and that's why you have two pulses. But in any case, what I want to show you is even this one is very interesting. There's no oscillation, but it's extremely dynamical. And for reasons that are selfish for me, I am very much interested in this process, and tomorrow I will spend some time on this process, okay, in which we have a single pulse, okay, and the dynamics which is associated with this. Okay, so I have given you examples of dynamical systems, of dynamical processes in cells. There are many, many more. I could spend hours. We could spend hours looking into the literature, okay? This is just, I took this from a slide by Mike White, who was a discoverer of oscillations in NF-kappa B. I saw this slide in one of his talks, and I copied it shamelessly. I did it in my way, okay? I did the same thing because I love it. Because here you have the period axis, and you have examples of different kinds of oscillations. I'm going back to oscillations. So you have oscillations in calcium signaling, in the nuclear localization of certain transcription factors, in this CMP, cyclic AMP signaling, whatever it is, in glycolysis, which is a fundamental metabolic process. You have the cell cycle, you have the NF-kappa B oscillations that I told you. This is vital in the formation of a vertebrae. I'm not going to enter into this, but this is vital for us to be organized as vertebrates, the segmentation. I'm not going to enter this. P53 you have already seen, which can also be periodic. As you will see maybe in a minute, and then we have circadian rhythms, and we also have seasonal rhythms, right? The trees are going up and down in a year. No, now I lose my leaf, then I get them again. So this is not one day. This is 1 year. The period is 1 year. Okay, so you have to imagine that. But I cannot stop thinking about this. A cell is a dynamical system that programmed within it contains huge numbers of clocks. And they have all to be interacting with each other. So you have oscillators, which are very widely varying in frequencies, but they're interacting with each other. You cannot help it because they are sharing the same machinery. And I think this is another understudied problem, in my opinion. What is the outcome of all this coupling, of all these oscillators with this wide range of periods? Okay. Anyway, I usually like to recommend this book by Howard Berg. I think it's a beautiful book. It's very short. I really recommend it. It's 40 or 50 pages. And if you're interested in random walks, in stochastic processes, I honestly recommend it to you. It's already 20 years old or 30 years old or something, 30 years old. But I put it because Howard Berg is a very well-known biologist. I think he was from Harvard or whatever, very well-known. The first sentence in the book, when I opened it because I liked the title and said, oh, as I was recommended, I read the book at the preface. The very first sentence in the edition that I have, at least, is this. That's the way the book starts. Biology is wet and dynamic. When I saw that, I saw this is. So I cite it every single time because a very well-known biologist starts his book with this sentence. It's wet, of course, because it's, but it's dynamic. Okay, that's why. Well, I think it's very important how you start a book. No, it was a dark and stormy night. So this is the first sentence of a book. Is very important. And this guy decided to start his book saying that biology is dynamic. I love it. I don't think that the biological community has really moving too much in that direction. There's a fraction of people working in this direction, but it's very hard. Okay, but that's what I wanted to tell you. Okay, oh my goodness. The final thing I want to say is not only about telling you how many dynamical processes there are. Are they useful for cells? Well, you can say that they are useful. I mean, circadian rhythms are useful because a prey, going back to population dynamics, is able to predict that the sun is going to come up. So I'm going to escape before the predator wakes up. Of course, the predator says it's better for me to wake up earlier. So again, it's also good for the predator to have a clock. So it's clear that the circadian rhythm is beneficial. It's clear that the cell cycle is beneficial because if you have to have division, it's better to have it programmed in your genome to make it reliable. But so there are, I mean, I have already answered this question, but let me show you one example by Galit Lahab again. This was on a later paper when she became a PI at the Harvard Medical School. She was still working again with P53. This tumor suppressor protein is very nice because he has this paper in Science a few years ago already, again, very old, but I think it's very nice. The title is P53 Dynamics, Control Cell Fate. So we have the word dynamics, which is beloved in our circles, in a Science paper, which is very relevant. And the idea is that, look, this is a figure from the paper. P53 detects all kinds of stresses. DNA damage, as I was telling you, lack of oxygen, some kind of heat shock, some kind of damage to the machinery that allows the cells to divide in two, spindles. Anyway, all these things activate p53. And p53 can do several things. Can induce cell cycle arrest to prevent cancers, for instance, because if you prevent the cell from dividing, it's not going to proliferate and form a tumor. Okay. It can induce DNA repair, fix the problem. It can induce apoptosis, which is suicide. The cell kills itself. That's another amazing biological process. It can induce senescence, which is not exactly the same as cell cycle arrest, but I don't know what the difference, the real differences are, honestly. Anyway, the interesting thing is how this protein can have this kind of behavior in which you can produce different kinds of things in response to different kinds of inputs. Okay. Now this paper, I'm going to oversimplify, but the main message is that they were looking at DNA damage and they were looking at two different outcomes, senescence or DNA repair. And what they saw is that the outcome was one or the other, depending on the dynamics of p53. If p53 was behaving in a pulsatile manner, they saw that this correlated with DNA repair. If p53 went to a fixed point, they saw that it correlated with senescence. So that was their claim. Their claim is the dynamics of p53 is important to control the fate of the cell. And that is why I think it is nice. I don't remember if they had a model in this paper, but Galit's group has developed models of this kind of oscillations for many years. And maybe we'll come back to this later. So anyway, let me continue. Summary so far. I hope this is not too much. Summary. The challenges I wanted to tell you. First of all, the interactions between genes and proteins are nonlinear. There are many, and cells are highly dynamic. Okay. So how do we solve this? And so far, we are in a course with a lot of mathematicians and physicists and engineers. I hope I haven't made a survey, but now I haven't talked about a single equation or anything. But my claim, and I present the same talk to biologists. I have no problem. I gave this talk to biologists. And here is when I convince them something I don't need to convince you, which is that in order to address these challenges, we can use math. Okay? Because with the theory of dynamical systems, we are very much used to dealing with nonlinear things. Maybe there are many, and of course there's dynamics. Okay? So I'm going to use a quote by a very well-known theoretical biologist. I'm going to use this quote now because I like it. So I'm going to show you a quote and let's read it together. Okay? This is by John Tyson. John Tyson is one of these guys who have made lots and lots of models of cell cycle. I don't know if he's worked on circadian rhythms, but for cell cycle, he's master, okay, of making mathematical models of the cell cycle. And a few years ago, 20 years ago, he published a little paper somewhere hidden, and he wrote this following quote on the use of mathematical modeling. Okay, he said, the situation is something like finding a jumble of jigsaw puzzle pieces in a paper bag. Imagine that you have a bag, and then you find a lot of puzzle pieces, okay? The ones that you have to build to fill a puzzle. Now, our job is to put the puzzle together, but first of all, we do not have the picture in the front of the box. We don't know what we are building in reality because we have some measurements, but we don't have the whole picture, okay? Second of all, we don't know if we have all the pieces in the bag, okay? Most surely we don't. There are lots of pieces missing, okay? And finally, we don't even have a table. In biology, in experimental biology, we don't have a table to put the things together. Nowadays, and Blai will tell me, synthetic biology provides a little bit of testing ground, which you can test these things. And the fact of the matter is this course is about systems and synthetic biology. Synthetic biology could be an experimental table. But the claim is, John Tyson's claim 20 years ago is that mathematical modeling provides a table. Okay? So that's what I would like to think. I very much like this quote. I always use it. Once I met John Tyson a few years ago in a conference, and I told him about this quote. I told him I always cite it, and he didn't remember writing it, which was kind of sad because I was so excited that I forgot. And I thought, oh. Anyway, I will keep citing it, but he forgot. So I could just it's written. I could just say it's mine, but the problem is it's already published, so I cannot claim it. But he forgot. So anyway. I'm going to now finalize this introductory part with another, a more recent picture, if you will, from John Tyson and Bela Novak. And I recommend this paper to you. It's a very nice review paper. Well, this is written for biologists. It appears in the journal Trends in Cell Biology, but I think everybody can read. I think it's useful for everybody. I honestly recommend it. I couldn't post it in the GitHub because I think it's behind a paywall, but I think nowadays I don't need to tell you how to go past paywalls. And if somebody wants a paper, just email me, okay? I recommend it. The main message is here. The main message that the Tyson and Novak tell us is the following. On the one hand, we have experimental information. On the one hand, we have information coming from molecular biology of which genes and proteins interact with each other. This particular case is an example of the cell cycle in the frog embryo, which is you have here a set of frog embryos, okay? So when the frog embryo is dividing and these cells go into, these are 4 cells, they start from 1, 2, 4, 8. This process is a dynamical system governed by these molecules, okay? So molecular biology is going to give you these maps that biologists have used a lot without really applying any rigorous intuition. Mathematics can do that, okay? And this is what John Tyson, for instance, is doing. Okay, he has lots of models. So you get cell biology properties and you get molecular biology properties. This means, okay, you can look at the period, you can look at how the period changes with different conditions, you can look at the cells, or you can look at proteins. So these are 2 different scales. And you use this information to come up with a molecular mechanism. Now, the first comment that they have is that you cannot extract, that's what I was telling you earlier, you cannot extract knowledge intuitively from this molecular mechanism. This is what they call the curse of complexity in that paper, okay? Because there are too many components and there are too many feedbacks. So when you have a lot of components and a lot of feedbacks, you don't have intuition. You have what they call the curse of complexity. So they say, okay, let's move on. Let's do some kinetic. Let's write down equations, which is what we're going to do next. Okay? And then they say, okay, the problem that we have there when you write down equations, and I'm jumping ahead, but anyway, when we draw equations, we have what they call the curse of dimensionality, which I'm sure that of course, this is a very well-known phrase in artificial intelligence. It means slightly different, but it's related. The curse of dimensionality, there are too many parameters, and we don't know. We don't know many of these parameters in vivo. We haven't measured them. We have a problem experimentally, so we don't know many of these parameters. So that's the other problem. So we cannot extract knowledge of cell biology just from the kinetic equations, okay? Which is what a modeler would do. I think this is a nice paper because if you go to some theoretical biology papers, and this was happening already in the 60s and 70s and 80s, and I think this is what made people not like too much theoretical biology, in my opinion, and we could discuss with Santiago, that I'm sure knows a lot about this, and many others. For many years, there was a journal, which is the Journal of Theoretical Biology, which you may have published, and there are very nice papers, and biologists don't believe anything. And this might be one of the problems because they always, whenever I show a paper, a work that I have done to a biologist, tell me, okay, how do you come with the parameter? Yes. Of course, we don't know many of the parameters. How do we solve this? Well, we cannot stick only with mathematics, with writing the equations and solving them in a computer or just doing some approximation to solve them analytically. We cannot stay here. This is where the dynamical system theory helps, because now we can go here. This is part of their figure. This is not me, okay? So we can have this. We can plot the state field. We can plot vector fields, and I think Thomas has an entry into this, but I think Imma already did. Okay, so we can plot, and Thomas will come, I guess, probably today. So we can plot the vector field in a state space, and then we can determine attractors, they call them repellers and transients, and all these type of things. And of course, this is what we can do here, right, in this room. And then you can draw bifurcation diagrams in which you plot how a variable depends on certain parameters. Okay, now here you're already varying parameters in a systematic manner. So if somebody tells you, how do you know what the parameter is? Well, at least I'm exploring state space, or a physicist would call this phase space. I think this is a difference, but anyway. I would explore, we can explore phase space, and of course, what can I tell you, Josep has been done this forever. So we do these kind of explorations, and now how do we finish? We relate this to real life, and real life is you look at signal response curves, which you can do experimentally, because I can start changing one chemical and seeing how this chemical affects the process. And that's the whole paradigm that they call a dynamical paradigm for molecular cell biology, okay? I think it's a nice position paper. Again, I think it's too late, in my opinion, because the community has already moved past this, but I think that as mathematicians or physicists, we have to, I think, in my opinion, is we have to fight against this huir hacia adelante. How do you call it? Huir hacia adelante. I don't know how to translate this in English. Run away. Okay, they're running away. I don't care. I don't know. I'm not, okay, come on. You haven't solved the problem. I mean, if you have a mathematical problem, you don't say, okay, I'm going to do the new one. No, I'm sorry. I'm being maybe too. Anyway, that's the situation. This is the paradigm that Tyson and Novak proposed. Okay, I've been talking too much, and now we're almost. So since this is a school, and I don't want to be, okay, I apologize. I don't want to be, how do you call it, condescending or treat you as simple, very young students or whatever. But since this was a school, we were supposed not to give talks all the time. So maybe interaction, and you're not talking much except for Stefano and Mark, and of course the faculty. So let's see if we interact a bit. Okay, so now what I'm going to do for the rest of the talk, and then I will stop, and definitely I'm late, but I will try to compensate tomorrow and on Thursday. What I'm going to, let's spend the final minutes using some kind of, this is an experiment. I don't know how is this going to work. So I am going to ask you the following. So the people, I'm looking at the camera. I hope that the people in Zoom, I'm going to ask them to either go to the Zoom chat. Sorry, the Zoom chat. I have to, oh my goodness. I am going to send a file. Sorry, 1 second. Oh no, I cannot do it. Okay, I cannot do it because I thought that this computer was connected to Zoom. I apologize. I'm going to ask the people in Zoom to go to this, okay? And to download a file which is called, sorry, Brian, you know what I'm doing here. It's called Plickers registration, okay? The people online, not you. You don't need to, okay? Plickers registration. You download this from, and please let us know through the chat if you have problems. When you download the file, you will open the file, it's a PDF, and you will see a list of names. You click on the link next to your name, and I promise it's not malware, okay? You click on the link next to your name. The people here, we can do it in person. So I am going to do it. Unfortunately, the faculty, I'm not involving them, but anyway, Daria, is Daria here? No, no problem, I'm not done. Julia. Julia, and then we have Irmand, or Irmand, I don't know how the pronunciation is. And then we have Kevin. Kevin not here. I'm sorry. I don't go. Lorenzo. Okay. And now I have Mark that I know him. So Mark and Lorenzo. And then we have Marta. Martin, Natalie. Aha. And then Stefano. Abel. No. Abel is here. I thought you were online. You were online. Okay. Or no, maybe not. No, sorry. No, you were not online. I just misplaced you. Okay, so now you have one of these. The faculty doesn't have it, but I have a few more left if you want to play, but anyway. I only have 3, unfortunately. I'm sorry. I didn't put you on the list because I didn't want to be. You can share one. I'm sorry. And then the people online, I hope they are registered. I don't know. I can give also one to the boss. I'm sorry for Tomas. I don't have any. Well, sorry. Okay. So now what I am going to do is, for those of you who are here, you know how it works. You can imagine already. So this thing has 4 orientations. I'm going to ask a multiple choice question. I mean, I love this technology. The people who came up with this are amazing. I just want to show it to you so that you see how simple idea it is and how good it is. It's an amazing engineers. I love them. You see what this is? So it has 4 orientations. You can select A, B, C, or D, just putting it in the right orientation. You be careful with the orientation. It has a little A, B, C, or D on top. You see? So this is personalized for you. So it's just a QR code, but it's personalized for each person and personalized for the answer for this person, so they know who you are. I mean, it's an amazing. If you haven't seen it, if you have seen it, okay, sorry, I apologize, but if you haven't seen it, I like it so much. So what I'm going to do in the next 8 minutes or less is I am going to enter in here, and I'm going to ask a couple of questions. They're very easy questions today. Eh? So, but anyway. Oh, sorry. Hybrid. Oh, what happened? Oh, sorry. There is some problem here. How does this work? Ah. Oh. How does this work? Why can I support? No sé si la tengo. Ah. Display. Screen mirroring. Yes, thank you, Josep. Mirror. Gracias. Okay. Now, I am going to ask you to answer this question. Those of you who are in Zoom, if you go to the, oh, sorry. Oh, my goodness. I apologize. You have to go, those of you who are in Zoom, hope you can see this. The Zoom people, please, Josep, I hope you can. The Zoom people have to go to this website. This is the hybrid thing. I'm sorry, I'm trying to make this thing work, and I'm losing time, I know, but since I've been talking so much. The Zoom people go to my.plickers.com, and you should see when you go into the website that it's already waiting for me to ask a question, if you have registered already, okay? So first you register using the PDF, then you click here, and then it should be waiting for you. The rest of you, you are just looking. So please remember the people online, my.plickers.com. Okay, and then I'm just going to ask you to answer this question. Let me see if I can. How does this work? Oops. I don't know. Sorry, one. Here. I show response. Oh my goodness. Sorry. I apologize. I apologize. I knew this was going to happen. It's just a learning process. I need to unmirror because this thing is not working well. How can I? Is there a way that I can? Is there a way that I can? Ah! Oops. How does this work? If I do this, there we go. Okay. So now I am going to ask you to choose your answer and put the right thing right now. And then this means that I am going to record. So now the people who are online, okay, but you have, so Nicole has already answered. Can you just show me? Can you just show me? Okay, so it detects automatically. I just love it. I cannot just tell you enough. And I think Natalie is too small. Yeah, it worked. Wow. It detects immediately, and I would love that you see the, I'm sorry, I would love you to see the augmented reality view that I have. I don't know if any, Irman, oh, sorry. You have to take away your fingers from the figure because the finger was in the middle of the code. Yes, there we go. Okay, anybody that is missing? No, okay. Sorry, I know now who is attending the lecture or not, but this was not my intention. Okay, done? Okay, great. So what I'm going to do is I'm going to stop and I'm going to show the answer. Oh, no, I don't want to show the answer. I want to show the graph. Okay? I'm not showing names because I don't want to anybody feel bad, but I know that most of you answered proteins, so you were listening. This is a trivial question. I apologize. Tomorrow I'm going to ask more complicated questions, but just because before we finish in 2 minutes, I'm going to ask another one just for fun. So then this means, sorry, because I don't know what this is. I'm sorry. This thing is killing me right now. It did not work for me. Akil, oh, I apologize. Did you register? If somebody has problems, let me know. What's going on here? Oh, here. If somebody has problems today, let me know by email, and then we can try to fix them tomorrow. I'm sorry, Akil, and whoever else in the chat tells me that this is not working. Which have you tried? The people online, you have to try to register. Click on the link, and it has to say welcome, Akil, or welcome, Carlos, and whatever, and then you go to my.plickers.com. Okay? Okay, so I am just going to this one, mark done, and then I'm going to play this one. I really love this technology. I cannot help it because it's, and of course, I know immediately the answer if it's right or not, but that doesn't matter. We're just testing. Anybody else? Irmande. What is the? Where is? Irmande is here. Sorry, your fingers. Your fingers. Yeah, because you are covering partly the. No, yeah. Just put your. Yeah, just got the page from the top, if you will, from the top. Yeah. Okay. Okay, great. Anyway, tomorrow we'll do something more fun because this is just this is the typical thing I ask students to know if they were listening. This is not that's not what I went to do with you. But today I haven't explained anything serious. I'm sorry. After an hour and a half, I couldn't ask you anything very serious. So I apologize. Okay. So anyway, at least we know how this works, and tomorrow maybe we can do it again. So now I go here and I show graph. Ooh, most of you said over a factor of, sorry, I should have put here 10 to the 6, but anyway, I didn't, this is, I don't know, I'm using this for general audiences and I didn't want to put 10 to the 6. Anyway, so we have here 1,000,000, and most of you said 1,000,000, which means that you were listening, which is great. And I just wanted to finish here, just to emphasize the point that I don't know if you agree with me, but this is amazing, that cells have clocks with periods covering 6 orders of magnitude, and they are all coexisting together in cells. Now, in my opinion, it's a completely open, well, it's not completely open. There are already some papers in which, for instance, they try to study the coupling between the circadian clock and the cell cycle clock. If you look in Google, or I can send you papers, there are some papers by some groups, not too many, okay? But some papers are looking at 2 clocks, which, by the way, are very clocking periods, which of course is reasonable because if they are they're very close in frequencies, which is reasonable because then you could have an interest in dynamics and so forth. But in my opinion, it will also be interesting to see what happens with the faster clocks. How do they affect the? Maybe we can do some kind of averaging out things and maybe consider some kind of noise. I don't know. I mean, there is a lot of room for doing science on this, in my opinion, and that's why I wanted to end with this. So now we stop completely, and then when we come back tomorrow, I will start with a very quick overview of how to model these things, which eventually is going to end in the same type of things that Tomás, Imam might have been using so far. So I'm stopping, I think, if there is any question or comment. Otherwise, we continue tomorrow. Thank you. Nothing? No. They didn't clap. I think, no, you don't clap. You don't, no, don't clap. Don't clap. That's it? Okay, great. So see you tomorrow.